My name is Anais Gentilhomme, and that's really loud. I'm a first-year master's student at the University of Alaska Fairbanks, and today I'm presenting my preliminary results and my future methods, as this is the first year of my master's, in my research titled Predicting Optimal Growth Rates of Marine Bacteria Using Genetic Signatures of Cold Adaptation. So to give kind of a little bit of background before I kind of dive into my methods and results, I wanted to give a kind of a broad definition of the term of psychrophiles as organisms that grow at cold temperatures ranging from about negative 20 degrees Celsius to positive 20 degrees Celsius. And the research into these organisms is really broad and really interesting for many different reasons, particularly because 90% of our Earth's ocean volume is less than 5 degrees Celsius, including the Arctic Ocean, which is at risk right now with increasing atmospheric temperatures. But the primary result and the reason why we're all here at this conference in terms of astrobiology is that most of the water that we will find, or we have found in our solar system is solid ice. But in oceans such as on locations such as Europa are really likely to be cold. So looking at how these organisms thrive in these really extreme environments is kind of key. So I wanted to make a quick note before I get started. I wanted to say that most of the organisms that thrive at sub-zero temperatures also live and grow at warm temperatures. And these are known as psychrotolerants and psychrotrophics. As you can see with this figure, we have a kind of a broad range of strains that grow at a lot of different temperatures in different locations and different environments. And the blue kind of square represents the minimum temperature, the green circle is the optimal temperature, and the red is the maximum temperature. However, when we kind of reduce this figure down a little bit, we see that the true psychrophiles, or those organisms that thrive in that set range of temperatures underneath positive 20 degrees Celsius, lie in saline systems and like seawater and sea ice and marine sediment. And this makes it so that Arctic marine bacteria are really excellent isolates and proxies to study these kind of cold adaptive signatures and probably biosignatures that we could use and identify later on. However, I wanted to also note that a lot of these strains, as you saw in the previous figure, we have very few of them currently. And so in order to address that issue, we have isolated over 600 strains actually in marine bacteria and sequenced 50 of them. So this is a figure by Duclouzeau that has been unpublished currently, but it kind of shows the broad range of marine bacteria that we can find in sea ice. Unfortunately, psychrophily is not a phenotype that is really easily identifiable, which would make my job a lot easier. But as found in a previous study using machine learning by Feldbauer et al. In 2015, he found that actually, there it is, that psychrophily, which is kind of the orange triangle right there, has the lowest predictability out of all of the complex phenotypes that he looked at. Okay, so why do we do this and what is the purpose of this? Is that if this is possible, if we can use and predict psychrophily and use it to predict growth rates in Arctic marine bacteria, we could use it to make predictions in other uncultured taxa known only from metagenome assembled genomes or MAGs. These two figures are, that's cut off back down, are kind of examples, come back. These two figures are examples of MAGs, actually. So this is right here. This is known as a MAG of a psychrobacter. So all of these, this broad figure actually is when you compare a lot of different strains and compare them to each other, specifically their contigs, and the really closely bunched up together can be isolated into one strain. This is another figure of the same psychrobacter, so the little one in blue up there, of a MAG, but at a different assembly graph. So I got started in formulating my methods by looking at the mechanisms of cold adaptation that had already been determined. And these include osmoprotection through accumulation of compatible solutes and osmolytes, lipid profile changes for increased membrane fluidity, increased production of EPS, or extracellular polymerase substance, identification of cold shock proteins, and the identification of cold active enzymes. All of these, except for the cold active enzymes, are likely evolutionary mechanisms that are looked at either vertically or through horizontal gene transfer. The cold active enzyme has a vertically inherited change in amino acid usage, which I'll get to a little later. But with how I identified all of these mechanisms led me to kind of the creation of my goal and the so what, why am I doing this? And the goal, the end product of this study, if everything goes well, would be to create a phylogenetic informed predictive model to estimate growth temperature ranges and optimal growth rates using only gene content and amino acid usage. And so if this works, this could help to lay some groundwork for changes in a microbial community, structure and function in a really rapidly warming Arctic. Okay, and I also wanted to make a quick thing clear. You'll see in my methods that I sequenced multiple strains of the same species, and the reason why I did that and the reason why a lot of people do that is to look at the difference between the pangenome and the core genome. Most bacterial species, every new strain is going to have genes that have not been observed before, and the set of all of those genes is known as the pangenome. So the more strains of the same species you sequence, the more likely you are going to have that increase, that increase in genes right there. While if you just look at one strain, if you just sequence one strain of the species, you're only going to get a little bit of those genes and just the core genome. So it's really beneficial to sequence multiple strains. So no good research is complete without a prediction. And so I predicted that the requirement for increased protein flexibility, or the requirement to predict how psychrophilic these bacteria are, would be by looking at amino acid indices or ratios, which are publicly available through a lot of different publications currently. And so I am currently using these 5 amino acid indices because they were the one that was most currently on hand at the moment and the easiest to find. But all of these are really pretty straightforward, and they'll portray kind of a conventional statistic methods, but unfortunately science is not that easy, and I will also have to take into account what evolution provides, and in the sense of you're not going to have just straightforward, this is how the amino indices index kind of brought around, it's going to come this way, and then it's going to come that way. To start my methods, and so to create this model, I had to identify kind of a core or a proxy genus to create the model. And so I chose the genus of Coelia because this is a bacterial genus that is found in cold waters around the world, especially in the Arctic. But its members are mostly psychrophilic and halophilic, but primarily its genetic code actually reflects the geological evolution of polar regions. So this would help take care of my previous problem looking at the evolution of these amino acids. And so this is kind of step-by-step go-through of my methods. You start off with your observations, of course. Currently we have field collections from Utqiagvik in Barrow. 17 out of 17 from March 2012 have been sequenced. 1 out of 25 from April 2015 have been sequenced. That's currently in progress. Determining their phenotypic traits and determining how well they grow at various temperatures is also in progress. So right now I have about 19 different strains sitting in my fridges and freezers, and I'm looking at the temperature and how well they're growing at an optical density of 600 nanometers. I'm further doing the sequencing of all of these strains using DNA extraction, library construction, and annotation using the Patrick portal. In addition to doing primarily laboratory work, I'm also using 44 additional Coelia genomes that are downloaded from a public database. And with those and with my laboratory work, I was able to create a phylogenetic tree comparing all of those. I was also able to synthesize cold adaptation indices all from all of these genes and put them together and compare each. And these are figures that I will be showing momentarily. So once I have all of this data, the growth rates and the cold adaptive indices of amino acids, I'm going to take all that data and put it together into a phylogenetic generalized least squares regression of cold adaptation indices to create that model so that if you just give me the genomes, I can tell you what temperature it'll grow best at and how quickly it'll grow. So this is a heat map of the preliminary results. So at the very top right here, these are the Coelias that I'm currently studying. This is a heat map of all of the strains from a collaborator called Charles Sweet, which isolated several hundred strains of bacteria from Utqiagvik and Chesapeake Bay. And right now we have sequenced only 60 of them, and this is what we are presenting. Right here is a phylogenetic tree comparing all of these sequences. And the red represents how closely related they are, and the yellow represents if there was a fluke or even if they weren't closely related at all, actually. This is the big preliminary results at the end of my first year of my master's. So this is a histogram of all of the amino acid indices. They're labeled down here. I apologize that you cannot read them. Right here, these are all the Coelia strains. These top ones, the BRX, are the ones that I'm currently growing, while the rest of these are from public available databases. But you can see that these kind of histograms and curves are really closely related, and so the minor differences of the distribution of these amino acids were sequenced. The only real one is this one right here, which is kind of new, and I haven't quite defined why that is, so that's yet to come. But this really suggests that phylogeny in general, so large phylogeny, plays a more important role in determining these amino acid compositions than does the adaptation to local conditions, because these, oh my God, sorry. These are taken from Chesapeake Bay, but these are from public available databases. So these are taken from really a wide variety of environments and locations, and they still manage to have kind of the same structure, which is kind of incredible, in my opinion. So the next step, since this study is really in its infancy, is to complete the growth rates measurements of all the ones that I have sitting in my fridges and freezers, and sequence all of those strains. To catalog all these cold adaptation-related genes within each of the genomes, test for differences in adaptation indices of individual gene families, examine other genera that have more phenotypic or genotypic diversity. Primarily, if all of these are so similar to each other, it's going to become beneficial to use a different and apply a different genus. And right now, the one that I have on hand is most likely to be used is Psychrobacter. And finally, incorporate all of this data into a predictive model. And with that, I'd like to acknowledge my committee, my advisor, Dr. Eric Collins, Dr. Charles Sweet, Annelise Duclouseau, and Jody Deming and Shelley Carpenter for sample collection and helping me, and as well as my funding sources. And with that, I'll take questions.
Thanks. Do we have any questions for Anais? If you could come up to the microphone in the center and say where you're from and your name.
Hi, I'm Annalise Meyer. I'm from the University of Victoria. I just noticed something on your histograms.
Mm-hmm.
It looks like the histogram that was quite different from the other ones. It appears to actually be a bacteriophage, not
The phage one? Oh, yeah. Yeah, you're right.
So that would be.
That would be why. Thank you. Yes.
My name is Jana Braberg. I'm from Rutgers. This is very.
To the mic, please.
How much closer? So it's an interesting observation. So it is known, I'm a bioinformatician, and it's known in the field, we've done this data analysis multiple times, that things that reside, or bacteria that reside in cold temperatures or hot temperatures tend to share amino acid composition across the genes that are relevant for activity in these types of environments, right? The fact that they're evolutionarily similar bacteria tend to not have the same composition across everything, right, is actually expected. So there is a lot of horizontal gene transfer and adaptation, which is also driven by the environments. So I'm kind of curious, what is the big oomph, right, of these genes that are looking not like their genome, but similar across, even though the genomes are similar? Do you know what I'm saying?
I'm not quite understanding your question.
So
sorry.
yes, we know about the adaptation. So what is the big deal about them being different from the genome similarities? So how important is it that the genes that are useful for adaptation to a particular environment are different from the genomes that they come from?
It's important in the sense that they use different factions. I'm also admittedly new to the field, so this is probably something I'll take a closer look at and identify later on in my research. Oh, I'm sorry. Yes. So it'll be something that I take a closer look at. I'd love to chat with you actually later on if you have a moment to talk about that.
Yeah, absolutely.
Yeah.
So the big deal I was kind of looking forward to is if you identify the genes that are similar across a genome but different from everything else in terms of amino acid composition, you can identify new cold resistance genes.
Yes.
So I think that would be super.
That would be really cool.
So I think that's all the time we have for questions.
Great.
Thanks, Anais.